Okay.
Okay. Okay.
Okay, nice. Does it work? I speak. Yeah, okay. Hi, everyone. So I'm Cris. I will be talking to you a couple of times today. So let's go slow and calmly. Okay. Okay. Better? Can you listen? Okay, nice. So my first talk will be about Reads2Map, and I'm really happy that some people are using it already. So this is a collection of bioinformatic workflows that I started when I was a PhD in Brazil, in the University of São Paulo. And I'm improving it and expanding to Polyploids as a postdoc at Texas A&M. Sorry. Okay. So, and I started to work with that, and the motivation was because I was working with GBS. That's a software for building linkage maps in diploids, and it can also deal with populations that come from inbred lines, an outcrossing population, biallelic and multiallelic markers, codominant and dominant markers. And it's a software that was released in 2007. So it was released by former students in my past lab. And I was working with genotyping by sequence data, and I was having inflated linkage maps. As a master student, it was all big. And I was having like a recombination fraction matrix that looks okay. That had like the red diagonal, and the gradient was becoming like the cold colors, what means that the order was good. This is just a subset, so I would expect with 37% of the chromosome, I would have about 40 centimorgans, but I was having like 540, so it's a lot. And if I observe the haplotypes, then I see that this double recombination very often. That's not, we don't expect that for meiosis, so it was wrong. It was like, what is happening? And I was, not only me was having this issue, users from the entire world was sending me like, okay, play the linkage maps. And also at the same time, Marcelo was developing Poly, doing linkage maps for polyploid species. And he was also having the same issue with GBS. But luckily, we, MAPLAB and also MapPoly use hidden Markov model to estimate the genetic distances. And this approach has a possibility of you including an error rate for your markers. And before, one map had a very low error rate because it was created for dealing with wet lab markers like SSRs. And this error rate was very low. And then if I just adjust this, maybe taking the probabilities of the genotypes that comes with. Oh, they can't hear me. Oh, they lost my slides. Okay, so maybe it's in the computer. So I need to be in the computer. Here? It's the microphone. So I can't use the microphone probably.
Should you unmute it in the room?
So here's better, or here's better. Okay. Okay. So I will speak louder. Speak louder. Okay.
From there?
Project.
Project.
Project. Okay. Go back to presentation. Presentation mode. Yes. Maybe this one. Is it? Good. It's getting zoomed in. Here. Got it. Yeah. Okay. So, okay, coming back a little bit. So I was talking about the error probabilities that before was a small valid, and now we are trying to adapt to the ones that comes from the genotype column. So the PL, it's a parameter that comes in the BCF, like the format, and it's just the probabilities of each one of the genotypes. So instead of using a low error rate, we started to use these probabilities that come with the BCF. And this like shorten a little bit the group, but it's not yet what we expected. And that's for this example, it's just 40 centimorgans. But if I use like a global error rate that's a little bit higher, that's 5%, I can reach the size that I was expecting. And this means that I'm giving more flexibility to the Haydn Markov model to change the genotypes according to the chain, to the information of the chain. And that was nice. And then we implemented, oh, now we need to change. Okay, no spoilers. Okay. So then we implemented a function in OneMap that users could provide these probabilities. Like they can provide a global error rate, like low or high, as much as they want, or they can provide a probability for each one of the markers or each one of the genotypes. And good. And we did other updates in OneMap, and we released the OneMap 3.0. But the thing is, like, okay, we can allow the users to do that, but we didn't have a recommendation. Like, why? Like 5% is the best one. Using the other, maybe other type of genotype colors was the best. So we start to explore what comes before it, because the quality of the markers will depend a lot of the process of SNP calling and genotype calling. And then there's a lot of softwares and parameters involved. And like just the genotype calling part, like there's these 3 softwares that we tested, and they have different models, and they also consider different characteristics like allelic bias, dispersion, segmentation, and outliers. And the input for these softwares are the SNP calling softwares, the softwares that identify the SNPs, the DELs, and sometimes they can even do like multi-allelic markers, like the MNPs. So many softwares, and I didn't consider all possible. I picked some. So in this case was GATK, Freebase, Stacks, and Tassel. And other parameters can be changed also in the alignment, like in the quality of the alignment, and also depend of your data set, you have different results. So we could change also the reference genome and also the characteristics of the data set, like it's low def, higher def, how frequent is the enzyme, what's the genome species. So it's all different. And if we wanted to give recommendations also, we didn't have like a gold standard. So simulations would be useful. And I need to tell you, it's very difficult to simulate GBS. I couldn't reproduce all the errors, but the simulation was useful for testing some specific scenarios, like what happened if we have segregation distortion or not. And also to set some filters, because we identified some common errors that was easily eliminated by filters. And okay, but now it's many softwares involved, and it's FASTkey files are the inputs, and they are big. So these consume a lot of computational resources to test all the scenarios, and each one of the items there duplicate the amount of maps that we have at the end. And so it was really hard, and I needed to do this in different computers. And to be fair, I needed to keep all the versions of all the softwares in all the computers. So it was impossible to do only with R or Bash scripts. And then I figured out this nice workflow language. It's only one of them, like there's other possibles. I picked this one that's called WDL. It's from the Broad Institute. They help me to organize the scripts and the workflow. And it's good because organizing workflow, you can run the entire pipeline, or you can run pieces of it. And it solved the problem of the versions because it's connected with Docker images. So each one of the softwares have Docker images, and I can transport these easily from different computers. And it also helps me to manage the resources that I'm using. Okay. But yes, it was really cool, like figuring out the workflow language. For me, that was developing, but people that wanted to use that don't need to know the workflows. They just need to go to the Reads2Map page, the releases page, and get the latest one, and download the WDL, that's the workflow file, the zip file, that's the dependencies, and the input file template. And there are lots of possibilities for input. And we have like the template, but what you need to do is fill each one of the items with your files and with the choices that you want to make for analysis. It's basically like if you want to run stacks, if you don't want to run stacks, if you want to run 3 days, if you don't want to run 3 days, and the path for your files, and the path for your reference genome, and the reference genome indexes. And there are many ways of running the workflows. You can run this on cloud, you can run this local, but I would not suggest if you don't have a lot of resources. And you can run this in high performance computing. And I made a tutorial for the Reads2Map that I talked more about how, like the hands-on, and I also talked about this in the computational session. But I suggest, I tested this a lot because I needed to run a lot of maps, and I have some suggestions of the software that it's easier to use for running the workflows. And 2 suggestions is the Puma and the Kappa. And if you want to use cloud and want to have a graphical interface just to submit and run, Broad Institute also put together an interface that's the Terrabuild. That looks nice too. I split the workflows in 2 parts. That's empirical SNP calling and empirical maps. The idea is that the first part, you can run with the complete data set, and the input files, FASTkey files. And because it's GATK and Freebase are implemented, you can run this, doesn't need to be GBS, could be any type of sequencing data. But tassel stacks need to be GBS and can be any type of population. And this will generate DCF files at the end, 1 or more, depending on the softwares that you chose to run. And the second part, I suggest to run with just a subset because it takes a long time. And you can take like a chromosome of a piece of a chromosome, and the input are the VCFs. So you just say like, okay, I want this piece of that chromosome, and we run only on that, and can be many VCFs. So it doesn't need to be the VCFs coming from the SNP calling from the Reads2Map, can be from the other softwares too. And this is exclusive for F1 mapping populations. And this will be the maps with all possible pipelines. And the output will be a compressed file, a targz, that's input for a Shiny app, because I like to do Shiny apps. And the Shiny app will display many graphics that will help you to diagnose the best pipeline. And then once you use the Shiny app, you can go back to the VCF and build your linkage map with just that data set. And I have some results for deploys. So I ran all these softwares for 2 different data sets. One is aspirin, and the other one is roses. And they have very different depth, and they are different species. Genome size is different, and the enzyme is also different. So the idea is that I ran using different parameters, different global errors, different softwares, and I picked the one that gave me the distance between each one of the markers, that the y-axis that's smaller, closer to expected, and I looked to other types of graphics to show me if we have like these big spaces between markers, and then I need to see also the recombination fraction matrix to see if it's all fine. And then once I say like, okay, these parameters are good, this software are good, then I can build my linkage map with using this and R. So good, this is results for Dploids, and we publish it. So if you want to know more, just go to the paper. Because here's a polyploid workshop, so we want to talk about polyploids. And because I already have all the structure, the only thing that I needed to do was change the software at the end. So I recently included MapPoly instead of OneMap. And the idea is the same. So the Reads2Map gets the FASTQ files, generates a bunch of VCF files, like the kind of parameters that you choose. And if you run empirical maps, you also generate the compressed file for the app. And then in the app, you can select the VCF and go to MapPoly. And I created another tab for evaluating the polyploid data. And I ran with our roses data. We have GBS, we have like many data sets for testing. And here I'm showing just two. Okay, I'm still, yes, I'm already, okay, it's fine. So I have this data set that has, it's an auto-test product roses, is a GBS, the NGO is the enzyme used, and the parents, and I have a median depth of 107.4. And this time, so the shiny apple, it's interactive, so you can select only one of the pipelines or all of them at the same time. All of them will be a little bit messy, as you can see. And this time, we thought that maybe we don't build a single map for each one of the pipelines. We can build like 500, but with resampling 100 of each one of the pipelines. And we decided to do that because of the hiding Markov model and the multi-point approach. If we have a single marker that's bad, it will mess up with all the distances. So if we resample, I have at the beginning like 200 markers about for each one of the chromosomes, 2,000 markers for each one of the chromosomes. So I can resample 100 many times, and I give chances to leave the bad markers out, and I will pick the method that provide me more good maps with these 500 resamples of 100 markers. And to be able to do this amount of markers, I separate the markers for each one of the parents, so I can run it fast. And the result is this density curves. So in your left, you are seeing the map size for each one of these maps, these 500 maps that I gave resampling. And the red dashed line is 100 centimorgans. So I would expect that the peak, the good pipeline will have the peak close to this dashed line. And in your right, you will see the number of markers and also a density curve. And because what happens is that when I'm building the linkage maps, each one of these 500 linkage maps, the hiding Markov model also drops markers. So I want a pipeline that keeps the highest amount of markers in the map. So I want this peak close to the red, and that peak closer to the right corner. And the boxes, it's because the two top ones, it's using genotype probabilities from the softwares, and the bottom ones are using 5% of global waiver. Yes, possible to understand? Okay. But I put it all here. It's a mess. So to be able to see better, the ones that I considered that was the best, I was the combination of freebase and Tasso and Polyred. And comparing to the Dploid data sets, these results was really different. So a funny story is that before I implemented that, so as I don't know if you remember, but the other slides, the stacks and updog was the best ones for Dploids. And I didn't have these results, and I told our PhD students that just did the GBS, like, okay, just run stacks and updog. And if you see, and we didn't have good maps, it was like, oh, what happened with the data? But for the polyploid data, the stacks was one of the words. Stacks with updog. Stacks with updog, the purple one. So it was not a good one. So I shouldn't put a graphic about that, but I just came up now. Oh, okay. I get excited and then okay. Okay. So yes, so what I was saying is that it's different, the results comparing to the diploid ones. But the best ones for, in this case, I consider that was these ones, but I would not see, I would not just take conclusions seeing only the map size and the number of markers. It's important to see also the recombination fraction matrix, if it makes sense, if it has the diagonal as we expect, and also the doses and the segregation distortion, if it makes sense. But the other tab of the app can provide this. So then you select the pipeline that you want to see the result and display for you. And also the other idea was that, okay, we have some maps here that have the peak close to the 100%. Why we not like take a window like this, like close to the 100 and get the markers that are common to all of these maps that's close to 100, because probably they are good markers. And so this range is to select the size of the maps and then gets all the markers in that window. And you can download the marker ID of all these markers that's supposed to be good. And the app you can also build a map, but it's important to consider that this is not the end map. You should go to R and do this manually. And this is an example of why, because even selecting these markers that's supposed to be good in the peak, we still have like outliers. Like we have, I don't know if my mouse shows, but in the graphic there, you can see that there's a blue line in the middle. This means that that marker should not supposed to be there. So they needed to be removed in further analysis. But you can see a map and the distance and the recombination fraction matrix for that markers that you selected. And just to see if it would change, we've seen same species and same person doing the GBS and just changing the depth. We ran with another data set of roses, and we figured out that we didn't need to run 500. With this time, we ran only 100. And then we had this big peak of stacks and updog. And then I thought like, oh, maybe sometimes stacks and updog is good. But what happened is that actually the filters, I think I forgot to explain this graphic. So this is showing the number of markers that are dropped by filters. So the first column here is the raw data for one chromosome. And then I filter by missing data of 25%. And then I filter by segregation distortion. So in this case, stacks is dropping off like a lot of markers and doesn't give much space for the resampling to work. So that's why that peak so sharp. So comparing to the other ones, stacks are the two bottom lines. So it's the one that most discarded markers. So if you see, that's why it's a very sharp peak. And if you try to see the recombination fraction matrix for this combination, it was not good. And then was like, oh no, it's better to go with another pipeline. And then I selected the two others that was good, and then was tassel with polyred and tassel with updog. And the data sets that I ran showed that in general, the running updog and polyred is a good idea for polyploids. This is the recombination fraction matrix for that combination, tassel and updog. And the same thing, like selecting the markers only in the peak and building the linkage map. So I think, yes, good. And so I wanted to highlight again that this tool is not for building the linkage map, but it's for selecting the pipeline for you to do that. So building a linkage map, there's a lot of details that you need to change and you need to check before going further. So I would suggest to not do this automatically, like this workflow do. And I don't have a specific answer. I wanted to have like a recommendation, use just this parameter, use just this software, but it changes. Like the workflow, it's very stable because I'm controlling all the versions. And just changing this data set gives me different results. And so I don't have a single suggestion for all the data sets. So, but I do have like some things that, okay, maybe this one you don't need to test. And so I became like these ones I turn as default values in the workflows. And so if in the template of the inputs, you will see that some of them have a default. So that's why I call like the data set independent. That probably will not change for your data set. And so I try to do that to reduce the number of tests that users need to do. And you don't need to know WDL to run it. For just run a bash script acknowledgement, it's important. For if you want to modify, if you want to like test other parameters that it's not in the input file, you will need to know bash script or R or Python, depending on the task that you want to change. And you only need to know WDL if you want to implement new features, if you want to implement new softwares, then it's important to know. And I think also that Reads2Map can be useful to developers that want to test the new features or want to test if the software is still doing what's supposed to do after a change. Because I just changed the version of the image with that software and it can rerun the data set. So I think it can be useful too. So that's all. I would like to thank you all the authors from different institutions, and especially Texas A&M and the University of São Paulo, and also NC State that Marcelo is giving me a lot of input. And the project members, the collaborators, and I'm open to questions now, but if you have future questions or technical issues,